Okay, I'm gonna try to make this shorter than how my videos usually are. But starting off, we have the electron transport chain here, describing the events that take place. So firstly, from the Krebs cycle, we have NADH and FADH2 here. And these molecules, 10 of these and 2 of these, are used by these proteins that funnel these hydrogen ions from the matrix to the intermembrane space. These are used so they're reduced and they push hydrogen ions up here. So the intermembrane space is chock full of these hydrogen ions, and so there's not that many on the side. Um, in the same process, these electrons over here that are passed through the phospholipid bilayer and they go all the way over here where eventually they're reduced once they become water over here. But before we get there, these cytochromes push about, I think, 4 hydrogen ions at a time to the intermembrane space. Then eventually, when this space is full of hydrogen ions, naturally, it wants to go back to an area of low pressure or where there's not as many hydrogen ions. So they funnel all the way through one big proton or one big protein over here called the proton pump. And this pump, all these hydrogen ions going through this pump generates energy, converting ADP and a phosphate ion back into ATP. So that's where we get our 30 to 32 ATP from the electron transport chain. And so now we have more hydrogen ions on this side. And so we're able to keep those over here. We have the electrons from earlier. And with just some oxygen that's floating around, we're able to also convert this into water. So that's the electron transport chain onto action potentials. Um, I realize this diagram is not entirely anatomically accurate because all of these events occur in the nodes of Van Devere, whereas these just propagate the electrical current along the axon. But I just chose to do it like this because cramming all of this into this small space would be just difficult to read. So membrane potential changes happens at the nodes, not the myelin sheaths. So starting off with the axons, the EPSP, um, it's this excitatory potential from the presynaptic neuron. So in this case, this would be the postsynaptic neuron, and via spatial or temporal summation from multiple neurons or one neuron, we get small action potentials coming through here, and eventually, if there's enough going to the dendrites, um, then this soma builds up a gradient potential, and if it hits a certain threshold, I think negative 55 millivolts, if I remember correctly, um, an official action potential for this neuron is generated at the axon hillock, and that's when we switch from the gradient potential to the all or nothing, or like the on or off potential. So once we have this potential here, it goes through the axon, and this is where it gets a little complicated to explain, but basically this area, this would be in a node, as I said before. This would just be resting membrane potential. So it's more negative on the inside and a little bit more positive on the outside. This would be an example of depolarization, where it's going up to about positive 30 millivolts because of all the sodium going in, making the intercellular membrane of this myelin sheath more positive. And so the action potential in this case would be here if it went from this myelin sheath to this one. And so the action potential is here right now, but it was here earlier. And so this part of the neuron is in depolarization, or approaching hyperpolarization. So after there was an influx of sodium ions inside, then the channel switched, sodium channels closed, so the potassium channels opened, and all this potassium floods out of this node of Ranvier, and it makes the inside of the neuron more negative, and so it dips down to about negative 90-ish millivolts before the leaky channels are able to balance it back out to negative 70. So then it can be like this over here at resting. Yeah, and then action potential goes through here, it propagates, so it keeps on going like this. So after, let's say the action potential was here and then it goes here. Now that this action potential is here and it goes over here, then this area of the neuron would be under depolarization. So this would be purple, and then this over here would be red. This would be going through the repolarization and then hyperpolarization, and then this would finally be done going through hyperpolarization. So then this would be green again. This would be resting, eventually ready for a new all or nothing action potential once this neuron finishes and it receives another EPSP from a presynaptic neuron. And then over here, the action potential, it goes to the axon terminals, where then it's able to propagate through neurotransmitters to a postsynaptic neuron, using calcium to push, for example, acetylcholine through ex through man, what's it called? Exocytosis. There we go. Okay, and finally chloride shift. I tried to use as many colors as I could so it's visually pleasing. Starting with the regular chloride shift. So if we have a red blood cell inside of the plasma of blood, this is a systemic cell, so it's next to the tissue. So let's say after a muscle is used or something and CO2 is produced, CO2 goes through into the blood and goes into the plasma and also goes inside the red blood cell. Here, the CO2 combines with a hemoglobin and pairs over here, but the CO2 also goes over here and combines with water to form carbonic acid. Once carbonic acid is formed, one hydrogen dissociates and combines with hemoglobin to be stored over here. But when we lose this hydrogen ion, what we're left with is bicarbonate, which is, the buffer that keeps pH and things like that in balance. And this buffer leaves the red blood cell to enter the plasma, and to take its place, to keep that charge, we have chlorine here that is in the plasma and then enters the red blood cell. So this, um, after these components, the carbon dioxide and the hydrogen ions are traveling through the bloodstream in this red blood cell, and they finally get to the capillaries and the alveoli of the lungs. We have the reverse chloride shift, which is the same thing but in the opposite direction. So the chlorine that was inside the red blood cell enters the plasma. The bicarbonate that was in the plasma enters the red blood cell. Hydrogen ion that was with the hemoglobin dissociates, and this hydrogen ion combines with the bicarbonate to form our carbonic acid again. Carbonic acid dissociates into water, which stays here, and our CO2. We also have the CO2 from the hemoglobin pair that we had earlier. This associates as well, so we have more CO2. And then we have even more CO2 that was stored in the plasma. So one, two, three CO2s, one from the plasma, one from the hemoglobin, and one from the carbonic acid. All of these CO2s go out into the lungs to be exhaled for aerobic respiration. Tried to go over that a little bit quickly, so hopefully that was still good. And yeah, thank you for the opportunity.